Hello students, welcome to readmedprep Academy channel. Today in plant breeding part 1, we are going to talk about the economic botany, origin and history of agriculture and organic agriculture. What is economic botany? Economic botany is a study of the relationship between the people and economically important plants. It explores the ways by which humans use plants for food, medicines and other uses. Economic botany intersects many fields including established disciplines such as agronomy, anthropology, archaeology, chemistry, trade and commerce. From the very early times, human beings have coexisted with plants which played a vital role in their survival through a long process of trial and error. Our ancestors have selected hundreds of wild plants from the various parts of the world for specific use. The knowledge of the plants and its applications have led to the development of the humans and the civilization in many ways. Domestication of plants. What is this? It is the process of bringing plant species under the control of humans and gradually changing it through careful selection, genetic alteration and handling so that it is more useful to people. The domesticated species are renewable resources that have provided food and other benefits to humans. The possible changes that occur in the plant species due to domestication are adaptation to a greater diversity of environments and a wider geographical range, simultaneous or uniform flowering and fruiting, lack of shattering or scattering of the seeds, increased size of fruits and seeds, change from a perennial to annual habit, change in breeding system, increased yield, increased resistance for disease and pests, developing seedless parthenocarpic fruits, enhancing the color, appearance, palatability and nutritional composition. Here you can see some domesticated plants. Well, the wild maize, Diocinate, it was cultivated and domesticated to modern maize, Z maize, where it has reduced the majority of the metabolites, yielding more products and including those of amino acids, sugars and organic acids have been reduced in this. Increased phenolics. The wild rice was domesticated to harm the cultivated rice, where there is increased phenolics and polyamines. The wild wheat was domesticated to form emmer wheat or durum wheat, which has reduced phenolics. Example, the trichins, the algicins, the coumarides and the vanillins. Reduced amino acids and reduced unsaturated fatty acids. Wild barley was domesticated as cultivated barley with reduced phenolics, that is syringic acid, syringaldehyde and ferulic acid. Now let us move on to the origin and history of agriculture. Archaeological evidence of earliest record of agriculture is found in the Fertile Crescent region in and around the Tigris and Euphrates river valleys, approximately about 12,000 years ago. Here you can see in this image the map showing the Fertile Crescent around the Euphrates and the Tigris river. The earlier Greek and the Roman naturalists like the Theophrastus, Dioscorides, Pliny the Elder and Galen laid down the scientific foundation in understanding the origin and domestication of cultivated plants. In 1807, Alexander von Humboldt considered the original sources of most useful plants and their origin is an impenetrable secret. In 1868, Darwin's evolutionary theory proposed that origin of useful cultivated plants have existed through natural selection hybridization. In 1883, de Candolle, in his Origin of Cultivated Plants, studied 247 cultivated plant species and attempted to solve the mystery about the ancestral form, region of domestication and history. In 1887 to 1943, Nikolai Ivanovich Vavilov made an inventory of the diverse forms of our most important cultivated plants and their distribution based on the variety of facts obtained from morphology, anatomy, cytology, genetics and plant geography. Vavilov has given the center of diversity of a crop species which may be the center of origin for that species. Vavilov initially proposed 8 main geographic centers of origin originally in 1926. Later, in 1935, he named 11 centers of origin by dividing few centers into 2 and 3 centers and added a new center USA, thus making the 8 centers of origin into 12 centers. Here in this image, you can see the Vavilov centers of crop origin crops domesticated. Number 1 around China, 2 India, number 2A, number 3, number 4, which are in West Asia, number 5, number 6, number 7, number 8A, number 8B. In this table, you can see the value center of crop origin and what were the crops that were domesticated. 1. China. The crops domesticated: foxtail millet, soybean, bamboo, onion and crucifers. Number 2. India. Rice, sugarcane, mango, orange, eggplant, sesame. 2A. Southeast Asia. Rice, banana, coconut, clove, hemp. Number 3. Central East, which is wheat, pea, hemp, cotton, etc. were domesticated. Center 4. The Near East. Wheat, rye and many subtropical and tropical fruits. Number 5. Mediterranean center. Olive, vegetables, oil yielding plants and weeds. Number 6. Ethiopia, otherwise called Abyssinian, where the plants domesticated were wheat, barley, sesame, castor, coffee. Number 7. Mesoamerica, which includes South American and Central American center. The plants domesticated were maize, bean, sweet potato, papaya, guava and tobacco. The center number 8, South America, tomato, pineapple. 8A, Chile center. The domesticated plant is potato. 8B, the Brazilian, Paraguayan center. The domesticated plants: groundnut, cashew nut, pineapple, peppers and rubber. In 1968, Nihole Zhukovsky put forward the concept of mega-gene center for the origin of cultivated plants. He divided the whole world into 12 mega-gene centers. In 1971, according to Jack Rodney Harlan, agriculture originated independently in 3 different areas in different times or simultaneously. Hence, a crop may not have a single center of origin. Harlan says that the center of crop plant means the places of agricultural origin of the crop plants. The non-center denotes the place where the agriculture of the crop was introduced and spread. Thus, center and non-center interact with each other. Now, let us move on to the organic culture. What is meant by organic farming? Organic farming is an alternative agricultural system which originated early in the 20th century in reaction to rapidly changing farming practices. It is a production system that sustains the health of the soils, ecosystems, and people. Organic farming relies on ecological processes, biodiversity, and cycles adapted to local conditions rather than the use of inputs with adverse effects. Here, the basic steps in organic farming is the production of vermicompost, production of green leaf manures, crop rotation, proper biological management of the lands, animal husbandry, biofertilizers, manufacturing and manufacturing of the manures. Biofertilizers. Biofertilizers are defined as preparations containing living cells or latent cells of efficient strains of microorganisms that help the crop plant's uptake of nutrients by their interactions in the rhizosphere when applied through seed or soil. Biofertilizers could be also called as microbial cultures, bioinoculants, bacterial inoculants or bacterial fertilizers. They are efficient in fixing nitrogen, solubilizing phosphate and decomposing cellulose. They are designed to improve the soil fertility, plant growth and also the number and biological activity of beneficial microorganisms in the soil. They are eco-friendly, organic, agro inputs and are more efficient and cost effective than chemical fertilizers. Here you can see the table where the biofertilizers are classified as nitrogen fixing biofertilizers, phosphate solubilizing biofertilizers, phosphate mobilizing biofertilizers, biofertilizer for micronutrients and plant growth promoting rhizobacteria. Nitrogen fixing biofertilizers are free living like Azotobacter, Clostridium, Anabina, Nostoc. Symbiotic, Rhizobium, Anabina and Azolla. Associative symbiotic, Azospirillum. Phosphate solubilizing biofertilizer like bacteria, Bacillus subtilis, Pseudomonas striata. Fungi, Penicillium and Aspergillus. Phosphate mobilizing biofertilizers, Arbuscular mycorrhizae like Glomus and Scutellospora. Ectomycorrhizae, example Amanita. Biofertilizer for micronutrients, silicate and zinc solubilizers, example Bacillus. Plant growth and promoting rhizobacteria, example Pseudomonas group got from Pseudomonas fluorescens. Now, Rhizobium. Biofertilizers containing Rhizobium bacteria are called Rhizobium biofertilizer culture. Symbiotic bacteria that reside inside the root nodules convert the atmospheric nitrogen into a bioavailable form to the plants. This nitrogen fixing bacterium, when applied to the soil, undergoes multiplication in billions and fixes the atmospheric nitrogen in the soil. Rhizobium is best suited for the paddy fields, which increase the yield by 15 to 40%. Here you can see the image where the root nodules having the Rhizobium bacteria and the cut section of the root nodule showing the bacteria. Azolla. Azolla is a free-floating water fern that fixes the atmospheric nitrogen in association with nitrogen-fixing blue-green algae Anabaena azolla. It is used as a biofertilizer for wetland rice cultivation and is known to contribute 40 to 60 kilo hectare per crop. The agronomic potential of Azolla is quite significant, particularly for increasing the yield of the rice crop as it quickly decomposes in soil. Here in this image you can see the Azolla in the paddy field, and the B image you can see the Azolla. Arbuscular mycorrhizae. Arbuscular mycorrhizae, otherwise shortly called AM, is formed by the symbiotic association between the Phycomycetes fungi and the angiosperm roots. They have the ability to dissolve the phosphates found in abundance in the soil. Here in this image you can see the hyphae growing around the roots and penetrating into the roots and forming arbuscules. Apart from increasing the availability of the phosphorus, arbuscular mycorrhizae provides necessary strength to resist disease, germs and unfavorable weather conditions. It also assures water availability. What are the benefits of arbuscular mycorrhizae colonization? When there is no colonization, the nutrient transfer from the soil to the roots is hampered, and there is decrease in the absorption of zinc, ammonium, water and phosphates, and there is also a phosphate depletion zone occurs. When arbuscular mycorrhizae colonization occurs, there is increased resistance to foliar pathogens, increased drought and salt tolerance, nutrient transfer via arbuscular mycorrhizae fungi. There is local resistance to root pathogens, increased resistance to heavy metal toxicity. Seaweed liquid fertilizer. Seaweed liquid fertilizer SLF contains cytokinin, gibberellins and auxin apart from macro and micronutrients. Most seaweed based fertilizers are made from kelp, brown algae, which grows to the length of 150 meters. Liquid seaweed fertilizer is not only organic but also eco-friendly. The alginates in the seaweed that reacts with the metals in the soil and form long cross-linked polymers in the soil. These polymers improve the crumbing in the soil, swell up and then they get wet, retain moisture for a long time. They are especially useful in organic gardening, which provides carbohydrates for plants. Seaweed has more than 70 minerals, vitamins and enzymes. It promotes vigorous growth of the plants. It improves the resistance of the plants to frost and disease. Seeds soaked in seaweed extract germinate much rapidly and develop a better root system. Now, biopesticides. Biopesticides are biologically based agents used for the control of plant pests. They are in high use due to their non-toxic, cheaper and eco-friendly characteristics as compared to the chemical or synthetic pesticides. Biopesticides have become an integral component of the pest management in terms of the environmental and health issues attributed to the use of chemicals in agriculture. Trichoderma species are free-living fungi that are common in the soil and the root ecosystem. They have been recognized as the biocontrol agent for the control of plant disease, ability to enhance root growth development, crop productivity, resistance to abiotic stress, uptake and use of nutrients. Here in this image you can see on the left side the Trichoderma fungi, the species being Trichoderma viridae, a potential biocontrol agent for the soil-borne diseases which is used as a biopesticide. Beauveria species is an entomopathogenic fungus that grows naturally in the soils throughout the world. It acts as a parasite on various arthropod species, causing white muscardine disease without affecting the plant health and growth. It also controls damping of tomato caused by Rhizoctonia solani. Here in this image you can see the left side upper image, the Beauveria species. On the right side you can see Beauveria species infected insect on the green plant. Below you can see the different types of insects like the house flies, bugs, ticks, and caterpillars and honeybees being infected with the fungi. What is green manuring? Green manuring is defined as the growing of green manure crops and use of these crops directly in the field of plowing. One of the main objectives of green manuring is to increase the content of the nitrogen in the soil. It also helps in improving the structure and the physical properties of the soil. The most important green manure crops are Crotalaria juncea, Tephrosia purpurea, Indigofera tinctoria. Here in this image you can see the plants which are used as green manure crops. Crotalaria juncea, Sesbania rostrata, cluster bean, cowpea, Sesbania aculeata. The green manuring can be practiced as green in situ manuring or green leaf manuring. Green in situ manuring refers to the growing of green manuring crops in the border rows or as intercrops along with the main crops. Example, sun hemp, cowpea, green gram, etc. Whereas green leaf manuring is the application of green leaves and twigs of trees, shrubs and plants growing in wastelands and field bunds. The important plant species useful for green leaf manure are Cassia fistula, Sesbania grandiflora, Azadirachta indica, Delonix regia, Pongamia pinnata, etc. Here in this image you can see the green leaf manure crops like the Cassia fistula, Sesbania grandiflora, Lucina leucocephala, Calotropis gigantea, Glacyrida sepium, Pongamia glabra, Delonis regia, Peltophorum ferruginum, and Azadirachta indica. Now, some of the Indian plant breeders: Dr. M.S. Swaminathan, he is a pioneer mutation breeder. Sir T.S. Venkatraman, an eminent sugarcane breeder. Dr. B.P. Pal, famous wheat breeder, developed superior disease-resistant varieties of wheat. Dr. K. Ramaya, eminent rice breeder, developed several high-yielding varieties of rice. N.G.P. Rao, an eminent sorghum breeder, developed world's first hybrid of sorghum, CSH1. C.T. Patel, who developed world's first cotton hybrid. Chaudhary Ramdhan, wheat breeder, who is famous for C591 variety of wheat, which made Punjab the wheat granary of India. So today in plant breeding part 1, we discussed about the economic botany, origin and history of agriculture and organic agriculture. So thank you. Kindly subscribe, like, share and comment our channel Read MedPrep Academy. Kindly register in our website www.readmedprepacademy.com for UG and PG MCQs. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.